Hello, my children. It is I, the great and the infamous Professor Science Person. Today, I have an exciting lecture for you all as we delve into the complex, in-depth role that proteins play within our bodies.
Pathetic. We have already discussed this in depth. My rich intellect should not be weighed down by such arbitrary restrictions.
Well, last time was more of the basics of protein structure. But don't forget, we're talking about the most essential part of life. We're going to need another session to really discuss why these proteins are so relevant.
Fine, I'll entertain this notion. You may carry on, peasant.
Excellent. So let's move on to a protein I'm sure you're all familiar with, hemoglobin. Here we can see a molecule of hemoglobin. Now, take notice of the fact that, as we mentioned last time, hemoglobin is made up of 4 polypeptide chains, specifically 2 alpha chains and 2 beta chains. The beta chains here are shown in cyan, and the alpha chains are shown in yellow. Now, if we zoom in a little bit, we can see a special molecule present in hemoglobin, which is known as heme. Now, there is a heme molecule bound to every subunit of the protein. So there's 4 heme molecules in total. Each heme molecule contains this atom at its center, which is an iron atom. This atom of iron is specifically made to bind to a molecule of oxygen. This allows hemoglobin to bind to 4 molecules of oxygen in total. With me so far?
Aye, Captain.
Oh, Gary, nice to see you've joined us.
Wait, Gary, what are you doing here? You were supposed to be rehearsing your performance. Would you take my place at my upcoming family reunion?
Oh, sorry. This just seemed really interesting, so I thought I'd drop by again. Hi, guys.
Damn it, Gary.
Oh, well, personally, I'm happy to see you join us again. So let's turn our attention back to hemoglobin. Specifically, watch what's going to happen when hemoglobin becomes loaded with oxygen.
Imposter!
Not exactly. You see, protein structure, as we mentioned last time, is maintained by a variety of different interactions between the various residues of the protein. However, when something like oxygen, for instance, binds to the protein, it is interacting with these amino acids. This moves them slightly and changes their interactions with other amino acids. This results in a domino effect that changes the whole protein structure. Thus, the cardinal rule. Whenever something binds to a protein, its structure shifts.
Sorry, this may sound a bit rude. Like, what you're saying is really interesting, but how is it relevant to the cell exactly?
Oh, don't worry, Caleb. It's always good to be skeptical. So, let's break these points down. Proteins, as we mentioned, consist of these really wacky three-dimensional structures that allows them to form what are known as binding pockets. A binding pocket is essentially a groove in the protein's overall three-dimensional structure. It consists of amino acid residues placed in very specific three-dimensional orientations to bind to a specific chemical of interest, what is known as the substrate. Because these amino acid residues are oriented so perfectly, it will bind to that chemical and nothing else, for the most part, as we'll mention later. Therefore, the binding between the protein and its substrate is both specific and strong. But because, as we mentioned, it is dependent specifically on the protein's structure, the binding between the protein and its substrate will no longer occur if the protein structure were to change. So, if another chemical were to bind to the protein, say another substrate on another binding pocket, then the protein will no longer be able to bind to the first substrate, and vice versa. If the first substrate were to bind to the protein, it can no longer bind to the second substrate. This creates logical systems that can be used by our cells in what are known as signaling pathways.
This is an interesting concept, but it eludes my grasp, much like a wandering spirit at the gates of purgatory.
Yeah, this feels kind of Dark Soulsy.
Well, I understand your confusion, so let's go over an example together so that we're all on the same page. With that, I would like to introduce you to the epidermal growth factor receptor pathway.
What?
Okay, so the EGF is a special signaling pathway used to tell our cells to grow and reproduce in response to specific conditions. Now, this is quite the complex pathway, so I don't want you to get bogged down with the specifics. Instead, I want you to understand and appreciate how proteins are operating within these larger, more complex systems. So, with that, let's start from the top with the cell membrane. The cell membrane is essentially the barrier of the cell, separating the inside from the outside of the cell. Within this membrane, we find proteins that are essentially piercing the membrane. Here is an example of one such protein, known as the epidermal growth factor receptor, or EGFR for short, because it is quite a mouthful. Now, this is obviously a simplified version of the protein. However, I am keeping things simple so that you don't have too much going on at once. Now, this receptor has a couple of things. It has a part of it, or a domain, that is sticking out of the cell, a part that is sticking inside of the cell, and a part that is lodged in the membrane. This is important for anchoring the receptor to make sure it stays in its current configuration and doesn't just move out or into the cell. Now, this receptor kind of chills, just moving around the membrane, doing nothing much, until epidermal growth factor, or EGF, is released into the environment. Now, the EGFR has a specific groove that can bind to EGF, like seen here. When it does, it changes its shape, subtly, but enough so that it can interact with another EGFR that is bound to EGF. These two will then interact together and bind to each other, forming what is known as a dimer. This dimer will serve as the active signaling complex and will tell the cell that EGF is present, so please reproduce.
See, so they are too weak to do what needs to be done on their own. Pfft, pathetic. I am so much more than these proteins. My feats far outweigh these pathetic proteins. For instance, I managed to...
Yeah, sorry, you're muted. I would give you more of a chance, but I straight up won't. So, back to our friend EGFR. Now that EGFR has formed its dimer, it can do something peculiarly interesting. It will speed up a chemical reaction. Now, we'll discuss the ability of proteins to do this later on, but for now, all you need to know is that certain proteins can essentially increase the rate of the chemical reaction far beyond what is normally expected. When EGFR forms this dimer, it will do the same and speed up a specific chemical reaction. In this case, adding a phosphate group to the other member of the pair. Thus, the EGFR molecules will phosphorylate each other, adding a phosphate group to the other member of the pair. The phosphate group is coming from a molecule known as adenosine triphosphate, or ATP, which is shown here. This isn't very important for our discussion for today, but it is especially important in the context of the cell, something that we'll cover in a future episode. Sound good?
Wait, didn't we mention this in our first session?
Yes, excellent memory. It's true that the molecule you see here is in fact adenine, the nitrogenous base found in DNA, and this here is in fact the sugar phosphate backbone of DNA. However, right now, this molecule is not serving as a building block for DNA, but rather as a molecule that can give a phosphate group to another molecule, specifically EGFR in this case.
Interesting. I thank you for the compliment, by the way, but I am a gamer after all.
Okay, so anyways, the phosphate group shown here is taken from ATP and given to the other EGFR protein of the dimer. This reaction, specifically taking a phosphate group and giving it to another molecule, is known as phosphorylation. This phosphorylation of the EGFR dimer changes the shape of the proteins enough so that it is now recognized by the binding pocket of another protein known as GRB2, which is shown here. When GRB2 binds to the EGFR dimer, or specifically the phosphorylated EGFR dimer, its shape changes so that it is now recognized by the binding pocket of a new protein, which is known as RAS, shown here. This RAS protein specifically is already bound to another substrate, which is known as guanosine diphosphate, or GDP. Here it is now shown here. This is also similar to the building blocks of DNA, because here you have the nitrogenous base, or that is guanine, and here you have the sugar phosphate backbone. This is also similar to ATP, except instead of a triphosphate or 3 phosphates, we have a diphosphate or 2 phosphates. When the RAS binds to GRB2, its shape changes so that it can no longer bind to GTP. Instead, it now binds to a molecule of guanosine triphosphate or GTP, which is just GTP with an extra phosphate. This binding with GTP also changes the shape of the protein so that it can now no longer bind to GRB2. Instead, it is released into the cell where it can trigger a number of things. One of these is known as the mitogen activating protein kinase cascade, or MAPK cascade, which is very important in triggering the downstream effects of the epidermal growth factor in order to change the cell's reproduction rate.
Excuse me, I think Bartholomew wants to say something.
Oh, right. Sorry, Bartholomew. What's up?
What do you mean by start? Is this only just the beginning of our journey?
Wow, I can't believe the great Bartholomew is taken back. Yes, RAS activation is just the start of the MAPK cascade. Essentially, this involves RAS adding a phosphate group to another protein. This activates it so that it can phosphorylate another protein, and so on and so forth, until eventually the protein that is phosphorylated doesn't just add another phosphate group to another protein, but instead changes the amount of protein that is produced from specific genes. This increased protein production changes the way the cell functions so that its reproduction rate is now increased. Thus, the EGF pathway has done its work in telling the cell to increase its growth rate.
What kind of an eldritch maze is this?
Well, there's a couple of reasons. First, you have to keep in mind that this pathway didn't simply poof into existence, but it came from living organisms, which in turn came from evolution. Evolution cannot simply create things out of nothing, but it can only tweak what is already there. Thus, these pathways tend to be very complicated to have many points along which evolution can tweak. Additionally, this pathway allows for the signal to be rapidly amplified. After all, a singular EGFR dimer can activate multiple RAS molecules, which in turn can phosphorylate multiple different proteins, and so on and so forth. This allows the signal to be rapidly amplified, allowing the cell to be very sensitive to the presence of these signaling molecules like EGF. This is very important because signaling molecules like EGF tend to only be produced in minute amounts. Thus, the cell has to be very sensitive and respond to any small increase in the concentration of these molecules. Furthermore, having so many actors involved in these signaling pathways allow for a lot of variation in these pathways.
And what does that mean?
Well, you see, we've been tackling the EGF signaling pathway as if it's a linear pathway, but in reality, it's anything but. Take EGFR, for instance. It is a receptor that is found in many different subtypes, with different subtypes found even on the same cell. It turns out that these subtypes have different specialities and can even form dimers with each other, meaning that different EGFR subtypes can form dimers to each other, in what are known as heterodimers. These heterodimers have distinct effects, which are still being studied. Additionally, the proteins involved in these signaling pathways, like GRB2 or RAS, have other functions in the cell, which are affected by these modifications, like the phosphorylation we mentioned, or binding to GTP instead of GTP, which will then affect their functions in the cell. Thus, the cell is impacted in a myriad of diverse ways, that there's not just the increased production of certain proteins.
I'm sorry, I'm completely lost.
Okay, that is completely understandable. Keep in mind, I'm not expecting you to learn any of the details of this pathway, like which proteins are doing what. Instead, I want you to take home a few key concepts about how proteins work in our bodies. Number one, proteins have super specific functions, and these functions are determined by their super specific and wacky structure. Number two, Proteins are able to form binding pockets, which are grooves that allow them to bind to a very specific molecule. Number 3, when proteins do bind to a molecule, it disrupts its structure and changes it. Number 4, like we mentioned, a protein's function is determined by its structure. So, if its structure is altered by binding to a molecule, its function will also change. And number 5, proteins are super duper important. Keep in mind, in this pathway, not only are they responsible for sensing the signal to begin with, but they are also responsible for carrying the signal inside the cell. And also, the increased production of proteins is what the signal is changing. In reality, proteins aren't just essential to life, but they are life itself. It is the proteins that are facilitating everything we need in order to stay alive.
Wow, so a bunch of proteins working together, each performing a relatively simple task, can carry out a complex function like cell signaling, right?
Wow, that's remarkably insightful, Caleb. Great job.
Oh, thank you, Professor.
No problem. However, there is something that you said that is worth addressing. You see, proteins may be much less complicated than whole cells or whole organisms. However, they are much more complex than what I let on during the session. For instance, take a look at this protein over here.
Oh, what is that?
Well, what you see before you is a small part of a protein known as p53, which has been dubbed the guardian of the genome. I'm bringing it up here because it is an excellent example of just how complicated proteins can be. You see, p53 is responsible for handling stress responses. When the cell is stressed due to any number of external factors, p53 is responsible for telling the cell what to do, and it has a number of different options. For instance, it can tell the cell to not replicate for a while, to sort of calm down, figure out what's going on before it continues with its normal growth cycle. However, p53 can also tell the cell to permanently halt replication, sort of preventing it from dividing and producing more cells in which the damage can spread. This is known as senescence. However, the most aggressive function p53 can take is actually apoptosis, which is literally cell suicide. If the cell is beyond any hope for repair, p53 will tell the cell to kill itself, to sort of destroy itself in a self-destruction sequence to prevent the cell from harming the organism as a whole. Now, with these multiple courses of action, p53 can take any number of them. Which course p53 decides to take ultimately depends on the context of the cell. Therefore, p53 can, in a way, decide what action to take by analyzing the context of the situation it's in, and thus decide on which outcome is best for the cell.
Ridiculous. Proteins do not have minds or souls. I tried to commune with my protein shake this morning. The result? The normies laughed at me.
Normies do be cringe.
See,
Well, okay, p53 cannot really decide the fate of the cell based on any inherent sense of logic or rationality, but it can decide the fate of the cell based on pure chemistry. p53, you see, interacts with many different proteins, which all respond to different aspects of cell stress and cellular conditions. The output of all these different interactions will then decide the course p53 wants to take for the cell. Here is an example. There are cells in our body that are actively reproducing at a very fast rate. These are known as stem cells, and their job is to produce the cells that are being lost by normal wear and tear. However, it is very important that these stem cells are free of any external stressors. Why? Because any damage to the stem cells, say in their DNA for instance, will then affect all the cells that they produce. Thus, damage to one stem cell will damage many cells. Therefore, these stem cells produce factors that tell p53 to be extra aggressive. If in another cell, it would just tell the cell to calm down and wait for the damage to blow over, in a stem cell, it might tell it to permanently stop growing or even kill itself. Therefore, it makes sure that the stem cells are free of any damage and are only producing healthy, viable cells. Now, if you excuse me, to be honest with you, I've been quite hungry all day and I've been waiting to eat this for quite a while.
Looking for this.
How did you do that?
That'll teach you for muting me. Know this. You may be the professor here, but I know secrets so deep that they will make the purest of hearts go dark with evil.
All right, fair enough. You do make a respectable opponent. I will admit that.
Indeed, I do.
Okay, and with that little exchange, I think we should end for today. Have a great day, guys, and stay tuned for our next lecture. See you!